In this section, I want to look at how males and females are epigenetically different from each other. But before I can do that, I need to explain what epigenetics is. Well, the prefix epi simply means over or above. So for instance, the epidermis of my skin lies over or above the dermis. This gives us an indication that epigenetics has something to do with something over or above DNA. And with that in mind, I'm now going to make an epigenetic modification to this DNA molecule I have here. But before I can do that, I need you to use your imagination a little bit. I need you to pretend that this little peg is a small chemical cluster known as a methyl group. Now, a methyl group is comprised of a carbon atom and 3 hydrogen atoms. So keep that in mind, and remember, what I'm doing here is not necessarily to scale. So now I'm going to make an epigenetic modification to DNA. Here we go. Okay, there we go. Epigenetics. Okay, so what have I done here? Well, there's a few things I want you to notice, in fact. I've obviously added a methyl group to DNA. Notice I haven't made any change, though, to the DNA sequence. Another thing I want you to notice is that I have deliberately added the methyl group to the DNA letter cytosine, because that is where DNA methyl groups tend to attach. And the important thing is here, even though I haven't changed the DNA sequence, changes like this to DNA have the potential to change the way DNA is expressed. In particular, if DNA is methylated adjacent to a gene, it has the potential to effectively turn the gene off or stop the gene being expressed. Alternatively, with the removal of methyl groups next to a gene, this has the potential to turn the gene back on again, facilitating gene expression. So DNA methylation is an important form of epigenetics. Now, there are other forms of epigenetics. Some relate to the way DNA is packaged, but DNA methylation is the one that I want to focus on here. Now, there's lots of things that can influence the methylation of your DNA, such as your diet or exercise or stress or exposure to certain toxins. So these things can influence the methylation of your DNA and therefore potentially influence the way your DNA is expressed. But with that in mind, I'm going to now put my DNA model to the side for a moment. So I'm going to get it out of the way, and this other stand I have here. And what I want to do now is I want to take a closer look at a specific example of epigenetics in humans. That's how it works at the DNA level, but how does it affect an entire person or an entire organism? And to do this, I want to look more closely at chromosome number 15. Now, by this stage, you've probably already realized that chromosomes come in pairs because we get one set of chromosomes from each of our parents. And so in my instance, I got one copy of chromosome 15 from my father and one copy of chromosome 15 from my mother. And so in this diagram here, to represent that, you'll see my 2 copies of chromosome 15. And you'll notice that I've coloured the one I received from my father, the ends of the chromosome, in blue, and the one I received from my mother, I've coloured the ends of the chromosome in pink. Now, I'm not implying that all women love pink and all men love blue. I'm just doing it so you can keep track of them, okay? So that's usually what we get. We get one copy of chromosome 15 from each of our parents. However, in rare instances, sometimes people can get 2 copies of a chromosome pair from one of their parents and none from the other. So if we go back to this diagram here, there are rare instances where someone can be born with 2 copies of chromosome 15 that they receive from their father, and they don't get any copies from their mother. Now, if this occurs, if someone's born with 2 copies of chromosome 15 and they're 2 paternal copies of the chromosome, they will get a serious genetic condition referred to as Angelman syndrome. Now, Angelman syndrome involves developmental delay, intellectual disability, and sometimes seizures. Alternatively, in rare instances, people could be born with 2 copies of chromosome 15 from their mother, 2 maternal copies of chromosome 15. In this instance, people develop another serious condition referred to as Prader-Willi syndrome. Now, Prader-Willi syndrome involves physical, intellectual, and behavioural problems. Now, this is intriguing. We're not necessarily talking about a mutated gene and important genetic information being corrupted. But what we have here is if we don't get a maternal and paternal set of chromosomes, we have these severe developmental conditions occurring. Now, the answer to this lies in epigenetics. In certain regions of chromosome 15, there are certain genes that are switched on and off because of DNA methylation in different but complementary ways. Angelman syndrome occurs because there's a gene which is usually only on the maternal chromosome. And so if you get two paternal chromosomes, that key gene won't be expressed and Angelman syndrome will result. Alternatively, Prader-Willi syndrome, there's certain genes, nearby genes, that are usually only switched on the paternal chromosome and they're off on the maternal chromosome. So if you end up getting two copies of the maternal chromosome, those key genes won't be expressed and Prader-Willi syndrome will result. So this phenomenon whereby certain genes are switched on and off in differing but complementary ways because of DNA methylation and they differ in males and females, this is referred to as imprinting. Now, there's like DNA methylation can occur throughout your genome, but when I'm referring to imprinting, I'm referring specifically to genes in males and females that are methylated and therefore switched on and off in differing but complementary ways. And when we consider humans, we have at least 100 imprinted genes, it would appear, in our genome. So this is how the geneticist Nessa Carey describes this phenomenon of genomic imprinting. Scientists have discovered that certain regions of DNA carry epigenetic modifications that indicate I'm from mother or I'm from father. This is known as a parent of origin effect. In these regions of the genome, normal development is critically dependent on inheriting one copy of a specific gene or genes maternally and the other paternally. So you see, that's why we need to get one set of chromosomes from our father and one set from our mother, because that will help us to get the balance of these imprinted genes where they're switched on and off in differing but complementary ways. This phenomenon of imprinted genes also explains why it's very difficult to create a living being from two sets of maternal or two sets of paternal chromosomes. Now, this hasn't stopped scientists from trying. So, for instance, back in the 1980s, a scientist at Cambridge University by the name of Azim Surani tried to breed mice that were made that had either two paternal chromosomes, so they effectively had two fathers, or either two maternal chromosomes, so they effectively had two mothers. And this is how one scientist described the result of that research. In the 1980s, Surani decided to investigate whether the genomes of two sperm or two eggs could develop normally when transplanted into an egg that had its own genome removed. If the two genomes were essentially the same, this ought to have resulted in the development of a normal embryo. Instead, Surani found that both combinations led to highly abnormal development. Now, by highly abnormal development, he's saying no mice survived, none were viable with these experiments. However, that was back in the 1980s. Since then, some things have actually changed. Back in 2004, scientists reported for the first time that mice had been created with two sets of maternal chromosomes, so that they effectively had two mothers. And more recently, scientists have announced that mice have been born with two sets of paternal chromosomes, so they effectively have two fathers. So this was summarized in a recent New Scientist article published in June 2025 with the title, "Mice with Two Fathers Have Their Own Offspring for the First Time." Now, here are some significant quotes from that article. The reason it is such a significant feat to create mammals with two fathers or two mothers is due to a phenomenon called imprinting, which is related to the fact that most animals have two sets of chromosomes, one inherited from the mother and one from the father. Crucially, epigenetic programming in mothers is different from that in fathers, with some genes that are labelled as on in sperm being labelled as off in eggs and vice versa. This means that if an egg has two sets of maternal chromosomes or two sets of paternal ones, it cannot develop normally. A gene that should be active in one chromosome of a pair may be turned off in both, or both copies of a gene may be active when only one should be, resulting in an overdose of that gene. Now, the way scientists managed to recently create, for instance, these mice with two genetic fathers was via a process known as epigenome editing. Now, as the name suggests, epigenome editing involves changing epigenetic marks, specifically the methylation of imprinted genes, to get the balance of gene expression right for healthy development to proceed. But this was a very technical task and the success rate was very low. Only 2 mice survived out of the 259 that were transferred, the 259 embryos that were transferred into female mice. But of course, the question arises, could this be done in humans? Could humans be born with either 2 sets of paternal chromosomes or 2 sets of maternal chromosomes? Well, this question was recently addressed by an Australian geneticist in a recent book that he wrote on epigenetics. This is what he had to say. Genomic imprinting means that a zygote needs a maternal genome and a paternal genome. Dreams of male couples using IVF to fuse sperm cells and insert them into a denucleated donor egg and thence into a surrogate mother who will give birth to a bouncing baby are unlikely to be realised. Similarly, fusing 2 unfertilised eggs is not going to work even if it were technically possible. So there will be no same-sex parenthood in humans in the foreseeable future. To do so would require modifying all or many of the 100+ imprinted genes, currently beyond our abilities and not allowed under current rules about editing the germline of humans. So even though we've seen mice born with 2 genetic fathers and 2 genetic mothers, this is not something we're likely to see in humans for the foreseeable future. And one point I want to leave you on here is to remind you that you obviously have 2 sets of chromosomes in most of your cells, but as you be aware, you only have 1 set in your reproductive cells. Males in their sperm, females in their eggs only have 1 set of chromosomes. And so the question arises, how does that set of chromosomes, does the DNA in that set of chromosomes get methylated? Well, Kat Arney answers this question when she writes this way. When germ cells, the precursors of eggs and sperm, are being made, any existing DNA methylation marks at imprinted genes get wiped off. Then new marks are put on, specific to whichever sex the cell finds themselves in. And so the cycle continues from generation to generation. So notice, if you are male, you put male-specific methylation marks at imprinted genes in your DNA. And if you are female, you put female-specific methylation marks at imprinted genes on your DNA. And so this is a profound difference between males and females. Males and females are different epigenetically. And so that's what we've seen in this section. We've seen how males and females are different epigenetically when we consider imprinted genes. And in the next section, I want to take a closer look at the question of how we define biological sex.